Hi everyone, in today's module we'll be talking about two strategies plants can use to form symbiotic relationships. The first strategy is through a symbiotic relationship with bacteria. Now, the one important thing to note here is that plants can form symbiotic relationships with bacteria in the soil, but in today's module, we'll be talking about in particular a very special symbiotic relationship that only involves a very special family of plants, and that's the legume family. So that includes peas, soybeans, clovers, alfalfa. These are the plants that you will typically see form the symbiotic relationship that we'll be talking about today. And of course, there is a special group of bacteria that will be of benefit in this type of relationship. So this is the Rhizobium genus, and this bacteria we often call Rhizobia. And when we find these two groups of organisms in the same locale, they tend to form a symbiotic relationship. And this relationship is one where we have some sort of legume and our Rhizobia. And since it's a symbiotic relationship, we do have some benefits that go to both groups. So Rhizobia is going to provide nitrogen to the legumes, so a nutrient source, a very important essential element for the legumes. And then in return, the legumes are going to give some sort of carbohydrates. So we have this really great symbiotic relationship, and we don't have time in this abbreviated semester to talk about nitrogen and exactly how the bacteria go about getting the nitrogen in a form that is usable to plants like legumes. They are providing nitrogen in the form of ammonia, but we won't have time to talk about that. But it's important to note that we have this very special symbiotic relationship with a very specialized group of legumes and Rhizobia. And when this bond is formed, when the symbiotic relationship gets formed in this group of plants, we see these interesting masses of cells. So the root nodules are important here. So we tend to see these they look like big masses of cells here. And we find these on the roots of plants that have developed this specialized relationship with bacteria. So they form this mass, and it's called root nodules, and we'll talk about how those get formed on the next slide. And when we get the formation of root nodules, it's actually an infection by the Rhizobia. And this starts by a secretion of signals and also recognizing signals, and so we get this interesting back and forth of signals being released and received by both of these groups. And what happens is that the host plant that will eventually be infected, to kick off this process, it secretes flavonoids. And flavonoids are a phytohormone, and they act as attractants for Rhizobia. So you can see here in this part of the figure, the plant has already established this root hair. It has released this flavonoid. Rhizobia are basically attracted to the signal after they have received it. And in turn, the Rhizobia release their own signal, and that induces this growth of this root hair to form this curl. So after we get this curl, we get this breakdown in the cell wall. So here in this panel, you can see that the Rhizobia found a way in through breaking down the cell wall, and we get this infection thread that carries out through the entire root hair. And from this point on, Rhizobia proliferate inside root hairs. They then are able to get released into the cytoplasm, as we see here. And we get more secretions from the bacteria that cause cells in the interior here and here in the cortex to divide and form like the early versions of a root nodule. So basically what we see are these kind of little vesicles here. They are essentially kind of budding off infection threads within this group of cells. And so they're membrane bound, they're clusters within these group of plant cells, and then they each of these will be infected with the rhizobia. And once we kind of get that infection, then we get more and more cellular divisions, and this ultimately forms that large root nodule. So all of this containing bacteria that have the ability to transform nitrogen that it gets from the soil into a usable form for the plants, and plants in return are providing energy sources for the bacteria in the form of carbohydrates. And a fun fact to all of this is that this kind of process, the way it starts off, is very specific to each species. So a particular legume species can produce a very specific flavonoid, or again, that phytohormone that gets released into the soil, and that acts as a recognition signal for rhizobia. And rhizobia species will each have their own signal that they send out to the plant, kind of inducing that curling formation in the early stages of this infection. So when we don't have the very specific, species of legume and species of rhizobia, then that root nodule formation doesn't happen. So we need that very specialized relationship of the particular species. The second strategies plants can use to kind of get a leg up in particular environments in terms of getting their necessary nutrients is by forming a symbiotic relationship with mycorrhizal fungi. And this is slightly different in terms of the way this relationship gets formed. It's different from with the bacteria. We don't get this huge transition of an infection and therefore kind of these interesting masses of cells formed. Instead, what we get, it's almost like an extension of the root network. So we get this massive extension basically because of this symbiotic relationship with the fungi. So sometimes we can see it really well if we were to dig up plants. Other times we might need some sort of specialized microscope to see the fungi present. But really, we get this major extension, and we can get the root surface area increasing by 10 to 1,000 fold. So this dramatically increases the amount of soil that can be used or scavenged for nutrients. And this is a very typical relationship that we see. So if we were to go out to the McLeish Field Station, we would find a lot of species out there would in fact be utilizing this type of symbiotic relationship in order to increase their ability to obtain nutrients. And if we were to see this association with our own eyes after we dig up the roots, we could see these hyphae. So all these the hyphae is basically the filamentous structure we see in both of these diagrams here. That is your signal that you have this fungal association. So it's this filamentous structure, it's the growth of the fungus, and in a close association with the roots. And there are two different ways that the fungal hyphae can form an association with the roots. One is mycorrhizae, which is on the left here. And this basically means with all of these filamentous structures, the hyphae, they kind of will be in here, but they won't be inside individual cells. They almost in a way kind of wrap around individual cells. But of course, we do have this large network surrounding the entire root. The second structure that we typically will see will be the arbuscular mycorrhizae. So this basically means that the fungal hyphae have entered the cells in the roots and formed these structures inside their root cells. They're kind of tree-like, as you can see. They branch off quite a bit. So again, this is another way to dramatically increase surface area, meaning that we are exchanging something in the surface area, whether it's obtaining nutrients or sugars from the plant. So these are the two differences in the structure that we can get in association with the fungal hyphae. What the hyphae will do is they release enzymes into the soil environment. This helps break down whatever macromolecules are in the soil. And usually when these macromolecules are broken down, it's usually into something that is usable by the plants. So that gets absorbed in the hyphae, and then that gets basically transported to the roots and then absorbed by the plants. In exchange, of course, the symbiotic relationship, there's a benefit for the fungal group as well. So the plants are going to release, some sort of group of sugars, simple sugars, as a way to help, increase the growth of the fungal hyphae. So of course, there's a beneficial relationship here, and it's one of the main strategies plants use to increase their nutrient uptake. So these are two really cool associations that plants use to increase their nutrient uptake in any given environment. But it's important to note that even though a plant can have a very specific signal to a very specific group of bacteria or fungus, that the plant can actually germinate and grow without these associations. And that does happen. And so in what environmental scenarios do you imagine these would be most helpful? Well, I would imagine in any environment that has low nutrient levels in the soil would lead to this type of associations. Sometimes we do see correlating with this soil moisture status. So in arid environments and places that I've worked at in the desert, they have really low moisture levels and as well as low nutrient levels. And so we will tend to see more of these really cool root nodules in that type of environment. Whereas something more around here, like McLeish, we might see more of these mycorrhizal associations. And what's really cool is that in 1 gram of soil, we can have up to 50,000 bacterial species, or even end up with, I've heard estimates of 200 meters of fungal hyphae. And that's really amazing. So they are there. So whether a plant uses them or not, we will find these groups present. But of course, as I mentioned, 50,000 bacterial species, that's a lot to, get into. And so, with, regular plant physiology, we can't quite get at that. If we collaborate with someone that has the means and the lab set up to actually get at who is who in the soil and the number of species, well, we can do a lot with that information in the future. That's it for today's module. Your main takeaway should be the 2 different strategies that plants can use to increase their nutrient uptake. First is through a symbiotic relationship with certain bacteria and certain plants in the formation of root nodules. And the second is by certain associations with fungal groups in order to increase the surface area in the roots to increase nutrient uptake.